Hi, everybody. Thanks so much for being here today. Thanks for John for his talk, which is very enlightening, and I look forward to following up with some details on some of the things that we've been up to in British Columbia, as well as with my colleagues Cindy Orser and Aaron Hilliard from DigiPath Labs in Las Vegas. Essentially, you'll notice that there's going to be a lot of parallels there with John's talk. So I think you've seen these illustrations of the plant morphology, and I like to refer to cannabis as essentially a single species with multiple gene pools. And as we all know, and what we've talked about a lot today, is the fact that the influence of cannabis on humans and vice versa has been nearly disproportionate in nature, in the sense that what you can see here on this current map is what I would say is the origin of different morphologies of cannabis, without referring to anything more than just the morphology and the size of the leaves. So you see narrow leaf hemp, NLH, kind of in Europe there, the broadleaf drug type, and the narrow leaf drug type, which those would be the indica sativa of the recreational or the medical market, and then the broadleaf hemp, which is more thought of as being the Chinese hemp. So essentially, these would be the kind of a primer of where the plants emerged and evolved. And essentially, as we know, it's moved around the world, and there's hybrids between most likely all of those gene pools at this point. Broadleaf and narrowleaf hemp have hybrids. And obviously, I think when we refer to drug-type cannabis these days, we're really referring to a large hybrid gene pool of both broadleaf drug type and narrowleaf drug type, so Afghanica and kind of South Asia origin, with some of the autoflowering traits and possibly specifically autoflowering traits originating from northern latitudes, so typically European hemp that was introgressed. But in essence, what we see is that we have a natural process of moving a plant around the world, and then we have the impact of artificial selection, like humans have had on any other crop. And here's a paper from Ernie Small, who essentially illustrates what this means. And so we have on the one side the fiber cultivars, and then we have more the narcotic cultivars. And the concept here is that there was divergent selection, selective pressures by the people that were essentially collected these plants and then selected them for very different reasons, whether, let's say broad categories of drug cultivars versus hemp cultivars. This relates to, this is the same paper, or another paper by Ernie Small as well, but this relates to the question everybody asks, hey, let's get, is it good to do commodity cannabinoids from hemp grown outdoors, or are we better off focusing on, growing indoor in a controlled environment, a drug type cannabis? So essentially what you see here and what this table shows is that there is a significant difference in between the size of trichomes in drug type cannabis versus fiber type cannabis. So hemp versus marijuana, there is here essentially almost a 1.6 times larger size of trichomes in drug type cannabis. So essentially you can see that this plant was purpose-bred for resin production. So essentially having larger trichomes, maybe more numerous trichomes, is also one of the key features there. And essentially for me, at this point in time, I think that's the large difference between hemp and cannabis, is really the, that's why we capped at 5% when we're talking hemp. That hasn't been introgressed with that key trait of resin production. I'm not going to go over the Solar paper, because obviously it was just covered, but what I did do here is essentially delve a little bit deeper into that concept of the divergence between these two gene pools, in particular European hemp and drug type cannabis. And this is work done recently, published at the end of 2017 by a group that I did my undergrad thesis and my master's thesis at the University of Lausanne in Switzerland, where I'm from. Essentially my master's advisor, Jérôme Goudet, and one of my committee members were the lead investigators on this study. And the interesting part about this study is that they used a type of genetic markers called single sequence repeat, or microsatellites. And the interesting part with those is that those markers are typically thought to not be under the effect of selection. So they're typically thought of being neutral. So it means that it's basically immune to the effect of people moving stuff around and essentially more so breeding for specific purposes. So neutral areas of the genome, they don't code for genes, but they might be associated with genes. So I think it makes more sense to use neutral markers when we're trying to decipher the evolutionary history of a particular, a particular structure that we're observing. And in this case here, we do see, as you can see here from the green plot on the right there, every single one of these bars is an individual plant. So there's 1,234 plants sampled here out of 48 populations. And I think this is probably the first population-level study on cannabis sativa where we have thousands of individuals and dozens of populations. So essentially red and green separates these two gene pools between hemp and drug-type cannabis. And then you see that there is essentially a clear pattern of structure, but there's not much more that we can see from this image. So thankfully, to these authors, they essentially shared their data. So I downloaded the data from the PLOS ONE website and repeated those analyses and added some essentially labels to it, but also going beyond that, seeing a particular structure where we have on the top right essentially the monoecious varieties of European hemp. And as we go kind of from the blue-colored to the yellow-colored, we're now going towards dioecious hemps. So essentially, monoecious hemp being a hermaphrodite variety, as opposed to the dioecious kind of being what's common for us to understand that we have both a male and a female plant. And it's interesting to see, and it makes sense there, that obviously the dioecious hemp cluster closer to the narrow-leaf drug type. And then we see essentially divergence towards the broadleaf, indica, afghanica, whatever we want to call it. But it really is quite interesting that we can tease apart a pattern here from a larger pool of samples than what had been seen previously using single nucleotide polymorphism, which are possibly both functional or under selection and also neutral. So I want to talk more about SNPs. John mentioned them. Essentially, this is what they look like here. It's a single pair difference on a sequence. It's one of the most common, sources of polymorphism. So essentially, the source of variation that's the most commonly found. It is typically thought to arise from a mistake in replication basically from one cell cycle to the next. And it's also one of the means of generating diversity, so using mutagens and things like that. But nature does it all on its own. It doesn't necessarily require us, and typically we see an average frequency of about 1 SNP for every 1,000 base pairs. And when we're talking about the cannabis genome, we see that there's actually quite a bit more SNPs, so large amount of diversity, about 1 SNP every 100 base pairs in cannabis. So essentially, what I want to talk about here, and this is an example from one of the licensed producers in Canada that I knew their horticulture director, and he came to me and said, something very interesting happened in one of our strains that we bought from the BC Bud Depot, which is probably not the most recognized seed source. But essentially, they have a variety called Shiatsu Kush, which they claim is from Japan, which it's not. But it's typically a high THC variety. And then here in this case, as you can see, something happened in the production line that caused one plant to express CBG almost in equal concentration to THCA acid. So it doesn't have to be a mutation. It could be something else. It could be an insertion of a transposable element right inside of an active THCA synthase gene. But it's really quite interesting how one of the reasons why they pick this up is because they have to QA, QC test their products. And when the one sample test comes back with a high CBG, they don't believe that it's happening, so they resend the same plant, and they see that actually something happened to one of the clones. And I'm happy to see that they're actually producing it as a plant and offering it as medicine to people that would be interested in having access to different minor cannabinoids. So these things can happen. If a mutation can happen in a small ACMPR greenhouse or a grow house in Whistler, Canada, it's definitely happening in those fields out there all the time. We're just not looking for it because of the scales we're growing at. So this is something that I did essentially using data from medicinal genomics, and part of the reason why I'm here is because of the philosophy of sharing this type of information with people to be able to, there's a lot of information. We can't all, go through it and decipher it. So here's an example, and I've got the NRC logo up here, and that's a little bit of a wave to John Page, because as you see, the USO31 and the phenol samples there came from them. So this is all in silico data downloaded from the internet. Again, thanks to Medicinal Genomics and John's group for sharing that data. Essentially what I do is use statistical approaches to be able to now decipher genomics and make it a little bit more approachable and maybe fast track some of these developments as we're talking about marker-assisted selection. So here you see that clustering, like we said, we've got phenol out there, hemp, and then we have the drug type cannabis that have some kind of a structure. But again, this is done with 28 accessions and starting with 180,000 data set with 180,000 SNPs. So what I did is in silico, I managed to use statistical analyses like discriminant analysis of principal component to reduce this 180,000 SNP data set to a set of 42 that in silico on the computer provided the exact same pattern. And here's something that I did also way back in the day, a 3D PCA that then was used by some other groups that are presenting their data in a fashion like this, but it's the same principle here. We see that there is a clustering going on, but again, a very basic structure that we see. So here's what I set out to do with another follow-up study in 2017 was essentially compare the indica sativa paradigm. So what I took data from the internet on terpene expression, and then what I did there is that I forced the software to say, okay, on the left here, I'm going to say indica sativa hybrid. How do they differentiate themselves? As you can see, they're all overlapping. And I tested a couple of options, including some of what the NAPRO research had done, which they said, I think it was limonene, caryophyllene, and myrcene. So I did that, and I did another, essentially, clustering possibility, which was myrcene, terpinolene, and limonene. As you can see, this was kind of what was the best cluster that I showed. And essentially what I did from that is I took the genetic data, constrained it based on these three classes, and was able to have a data set that almost perfectly clustered these groups into their relevant groups here. This is more of a proof of concept, but essentially this data is available on the internet. I have these 21 markers that are available for people to download and play with. And again, this is really just digested information from Cannapedia. So the study with terpenes and digipath really kind of came as a follow-up to that, and going from 70-something samples to now about 5,000 samples. And this is all terpene data, so you see all of the variables there, limonene, blah, blah, blah. And you see how essentially the software can cluster these groups into three different groups. And this is just drug type cannabis, but typically, a myrcene, beta-caryophyllene dominant in red, a terpinolene dominant group, and then a limonene dominant group with obviously some other terpenes in there. But we get a really consistent clustering of these three groups based on their chemotypic data. So what I did is the same kind of concept using fancy statistics to simplify and really actually generate synthetic variables called principal components. And here, what you can see is out of those 5,000 individuals, 70 samples that were sequenced essentially with a similar genotyping by sequencing approach by medicinal genomics, and you see that there's still that cluster still holds essentially, and there's some overlap there, and it's not a perfect fit, but essentially from that data set, I was able to generate 48 sets of primers, that I wanted to validate from the in silico on the computer studies to now being able to say we can do this in the lab in a repeatable fashion. So interestingly, out of 48 markers, we really only had about 23 that were validated, so 9 of them just never even amplified, so I think it's based on the large amount of SNPs in the priming areas of the primers. We found that 16 that showed polymorphism on in silico data were actually monomorphic, even between hemp and cannabis.
3 minutes.
Essentially, out of that, 18 of them were associated with terpene expression as well. And here's just an idea of the assignment where we compare the genome GBS, so 10,000 SNPs against 18 SNPs. You can see that the statistical assignment to the different groups is very, very close, with essentially 1,000 times less markers. And this has also been confirmed by some other groups. Essentially, this is a study out of New Mexico that shows the same kind of clustering with myrcene, terpinolene, and limonene. This is something that I validated with a licensed producer in British Columbia that I work with called the Flower Corporation, where essentially now we've taken, we see hemp here clustering by itself, and those are X59 and phenola samples, a myrcene dominant group. What we see here as the little cloud, the green cloud, that's actually a pinene dominant accession that has pinene and myrcene, but pinene is quite a bit higher. And you can see at the bottom, the pink is hemp samples, and so they're not all distributed in one part, but you can identify hemp very easily with this approach. I'm not sure what this is, I don't see it, but this is interesting. I'm showing the same data here, and what it shows is the markers that are associated with the clustering. So on the top left there is hemp, and we see mito, so it's a mitochondrial marker that's always fixed in hemp, and then SW6, which is actually a THC synthase marker that I developed that enable you to irrefutably identify hemp, so CBD hemp versus CBD drug type cannabis anyways. And then the same, when you look at hemp, there's different phenotypes in the fields that are also associated with the height and also the fragrance of the plants. And then again, the last thing that I did is essentially take this small set of markers, so actually just 23 of them, and I took all the data from Cannapedia, and I wanted to see how if we're able to identify strains. And again here, I identified a few of them, but one of the data that they show is a good clustering in a lot of the Blue Dream accessions, and the same with the Durban poison. So it's just nice to see that this actually translates across the board. And thank you so much, everybody, for the attention. Not much time for questions, but yeah.
Round of applause.